Like most other living organisms, bacteria evolve based on the conditions they are exposed to. The constant exposure to antibiotics led to bacteria developing drug-resistant mechanisms that would allow them to be unaffected by such drugs. E. coli is the most common gram-negative pathogens in humans because it can be found in a variety of foods and environmental sources, making it easy for people to become infected. As previously highlighted, the misuse of antibiotics has generated drug-resistant E. coli strains that can cause multiple different diseases, such as gastrointestinal and urinary infections, as well as severe pneumonia. Given the limited potential of antibiotics for combating drug-resistant bacteria now, scientists have been actively investigating alternative avenues to address bacterial illnesses. Bacteriophages are a favorable solution to this threat. These are bacteria-specific viruses that replicate by manipulating the metabolism of bacteria. They are the most abundant microorganisms in the biosphere and are more specific and even cheaper to engineer than antibiotics. Another excellent alternative to antibiotic is garlic. Garlic has long been known as the most effective plant species in treating bacterial infections. It contains several hydrophobic antimicrobial compounds such as allicin, vinylidins, adjuvants, and diallyl polysulfides. These compounds exhibit antimicrobial activity against gram-positive and gram-negative bacteria. It was then hypothesized that garlic would exhibit antimicrobial properties against E. coli with similar or even greater efficiency as an antibiotic, and that T4 bacteriophages could effectively infect and lyse E. coli bacterial cells. In order to test our hypothesis, two essays were done. The first essay was a bioplaque assay. This was performed to test E. coli susceptibility to T4 bacteriophages. So basically, a T4 bacteriophage stock was diluted into 8 tubes, each containing 9.9 mils of nutrient broth. And after mix, then 1 ml of the dilution was inserted in a tube that contained 3 mils of nutrient agar and 1 ml of E. coli. When mixed, then the content of that tube was poured in a plate with agar that was labeled with the corresponding dilution. The plates were then incubated overnight at 37 degrees Celsius. After overnight incubation, a search for evidence of plaquette-forming units in each plate was done. The second assay done was a disk diffusion test. This assay was performed to test E. coli susceptibility to garlic. 4 agar plates were divided into 4 quadrants. One quadrant was labeled as negative control, another one as positive control, and the last 2 quadrants were labeled as GAR1 and GAR2 for 2 garlic concentrations that were prepared. Next, 125 microliters of E. coli was inserted in the middle of the plate. Then it was spread. Antimicrobial discs were then inserted into each of the quadrants. The negative control quadrant received an antimicrobial disc that was saturated with sterile water. The positive control quadrant received an antimicrobial disc that was saturated with amoxicillin. The GAR1 quadrant received an antimicrobial disc that was saturated in 100% garlic concentration. And the GAR2 was the quadrant that received an antimicrobial disc that was saturated with 50% garlic concentration. When the treatment discs were in the corresponding quadrant, the plates were then incubated overnight at 37 degrees Celsius. The next day, the zones of inhibitions in the quadrants were measured in milliliters with a ruler to evaluate the results of the garlic treatments of the growth of bacteria. As the results section indicate, for the phage plaquet assay, there was no evidence of plaquet forming units observed in the plates. And in the case of the disc diffusion test, of the 4 plates prepared, only one was able to show the expected results. In conclusion, the experiment showed that garlic exhibit antimicrobial properties and the phage plaquet assay failed to produce plaquet-forming units. Nevertheless, this result can indicate one of the rare instances when bacteria possesses or develops several mechanisms to prevent viral infections. For future research, natural garlic should be used instead of processed garlic powder if greater zones of inhibition wish to be obtained.